Hi everyone, my name is Raina Bornstein. I'm 15 years old and I specialize in optogenetics. Optogenetics is an emerging neuroscientific technique which utilizes light and genetic engineering to control groups of neurons in the brain. The way that this works is there are a group of light-sensitive proteins called opsins, which are naturally occurring in plants such as algae. And in algae, the purpose of the opsins is for photosynthesis. So when activated by a certain color of light, many different colors of light come from the sun, and each opsin is receptive to a certain type of that light. For example, in the initial protein channel rhodopsin, which was first found to be able to perform optogenetics, it's sensitive to blue light. So when blue light activated the channel rhodopsin, then it would indicate to the rest of the plant that it was time to perform photosynthesis, and then the channels of the plant would open up in order to perform that. And in about 2009, then scientists realized that they could actually utilize the properties of these opsin proteins in the brain. So they injected certain neurons in the brain in a given group to edit them to express opsins, and then also put in the brain an invasive brain-computer interface, which delivered that specific color of light that the opsin was receptive to. And when they put the light on the neurons which had been genetically modified to express opsins, then they realized that the opsins would control the activity of those neurons, either by performing excitation, meaning that they would increase the activity of those neurons, or inhibition, which is when they would decrease the activity of those neurons. And they were able to do this because the opsins actually manipulated a process called action potentials. And what action potentials are when a neuron fires, it's because something called an ion channel receptor opens up, and on top of the ion channel receptor, there's a certain type of ions, for example, sodium. And on the bottom, there's a different type of ions, such as potassium. And when the sodium and potassium ions both stay on their respective sides and are entirely separate, then they are hyperpolarized and there's no firing in the cell. But when the ion channel receptor opens up, then it enables those different types of ions to mix. And when the ions become completely mixed, then that is when an action potential is generated. And then the neuron will fire, and that signal can spread to other neurons as well. So opsins, when they were genetically modified into neurons, would go onto the ion channel receptors. And when the specific type of light was shown onto those opsins, then the opsins would indicate to the ion channel receptor either to open up a lot more or to stay shut a lot more than usual. And that was how opsins were found to be able to control neuronal firing, which is the technique that we now know to be called optogenetics. I know this might just sound like a lot of scientific gibberish, but this actually is a very big deal, and the reason why is because optogenetics can be utilized to treat a variety of brain-based conditions. So when looking at excitation of neurons, excitation can be utilized for conditions such as neurodegenerative disorders. And those are conditions such as Parkinson's, Alzheimer's, ALS, Huntington's, and many more. They're called neurodegenerative because over time, as a person ages, then the ability of a certain group of neurons will degenerate, meaning get worse. And that impairs a certain group of a person's functions, which those neurons are used for. So take Alzheimer's, for example. It is a condition where the memory and cognition of a person are severely impaired. And the reason why is because Alzheimer's is based in the hippocampus and the VTA, which are both parts of the brain that control memory and cognition. So the ability of neurons in these two areas degenerates significantly over time, and the adverse effects that people with Alzheimer's have to face are direct effects of that because their memory and cognition become severely impaired. But by implementing excitation of neurons in those areas, by performing optogenetics, then you are able to force the activity levels of degenerating neurons in those areas to go back up. Optogenetics acts as a sort of forcing function to ensure that the ability of those neurons which had been degenerating is brought back. And then the effects of Alzheimer's are also combated because suddenly the patients who suffer from these effects have their neurons back in peak shape, and therefore their memory and cognition aren't impaired either. Then when looking at inhibition of neurons, that's useful for a variety of psychiatric conditions such as anxiety. So anxiety occurs based on too much connection between two areas of the brain, which are the medial prefrontal cortex and the basolateral amygdala. And by utilizing optogenetics to inhibit the channel ion receptors, then that will decrease the activity level between those two areas and therefore decrease the amount of worrying and fear that a person experiences, therefore combating the adverse effects of anxiety, which often are too much worrying or overthinking things, and those are combated by utilizing optogenetics in the context of inhibition. So by now, it's pretty clear that optogenetics is a great tool, right? And you can see how it could have such a strong positive impact. And one more thing that I'd like to point out is that yes, there are already pre-existing methods which can stimulate the brain, such as transcranial magnetic stimulation or deep brain stimulation, but the reason why optogenetics is so much better than these other methods in many senses is because when utilizing those other techniques, one activation in a certain area will activate every single neuron in the nearby area. But when using optogenetics, only neurons which have been genetically modified to express opsins will have any reaction to the light being directed at them. It's not harmful for any of the other neurons either. So you have really precise control over which neurons you're stimulating, and therefore the treatment is also far more precise. However, there is one big thing, one key factor, which is holding back the development of optogenetics. And this is the aspect of the system which involves an invasive brain-computer interface. And this is a problem for two reasons. One is more on the side of societal stigma, because invasive brain-computer interfaces are still very much not widely accepted in society. Many people are concerned about implementing these devices in people's brains, even in a medical context, because the reason why most people are afraid is kind of going into the ethics a bit. They are afraid that devices will extract personal information without a person's knowledge based on the fact that they are inside the person's brain at all times, in comparison to non-invasive brain-computer interfaces, which can be put on the person's scalp and then taken off when what they're being used for is completed. But since these devices will be inside the person's brain at all times, they're afraid that personal information will be extracted and potentially even exported to other sources without the person's knowledge or consent. So that's on the societal stigma side, but it's also holding back the development of the technology in a sense, because optogenetics, you may not know this, is still in the mice trials phase of its development. And I believe that part of that is because of the very unnatural evolution of the technique. It never has existed before in a non-invasive nature. And if you look at the evolution of brain-computer interfaces as a technology at all, non-invasive devices existed for a whopping 74 years before invasive technology even came into play. So it's clear that in the natural evolution of this technology, previously, there was a long period of time where non-invasive devices were utilized before the invasive aspect came into play. And there was never a non-invasive optogenetics, which means that people aren't even accepting of the technique at all yet. And this is stunning its development because it's much more challenging to get approval from the FDA and also even to develop this technology at all when there's no non-invasive baseline. So I realized that it was really important that there be a non-invasive baseline developed, at least for the time being, because that was the best version of optogenetics for today's society. And I decided to take that upon myself. So over the past month or so, I have been developing a concept for a prototype for non-invasive optogenetics, and that is what I'm here to tell you about today. This idea isn't entirely new because there are some models that have existed in the past for minimally invasive or semi-successful variations of non-invasive optogenetic models, and I took a look at those before I got into doing my own thing. And so the first example of a model of partially non-invasive optogenetics was the SOUL model. And it's called SOUL because it utilizes an opsin called SOUL. And the SOUL opsin is receptive to blue light, and what makes it really unique is that it doesn't require a lot of light to be activated. So that's why they were able to send the light in from a farther away point. However, it wasn't entirely non-invasive because the reason it was minimally invasive is because instead of being placed right on the brain or in the brain-computer interface was still inside of a person's head, but it was placed in a hole in the skull. A naturally occurring hole. There was no drilling required, but it was put in a hole in the skull. And so although it wasn't right on the person's brain or in the person's brain, it's clearly still invasive because it was in the person's head at all times. So that only partially achieved the goal. But then there was the carmine model. And carmine is an opsin which is receptive to red light. And red light, since it has a much longer wavelength and lower frequency, can more easily penetrate the brain versus any other type of light on the visible light spectrum. So although the opsin still required more light than SOUL per se, it was able to be non-invasive because unlike blue light, which is utilized in SOUL and cannot penetrate the skull too well, red light is able to do that really well. So they had the brain-computer interface device entirely outside of the skull to deliver the light. However, they were only able to penetrate as deep as 7 millimeters into the brain. And clearly, that is not enough. That's only about, this much. You can't even really see how much that is that well right now, but just think that it is not nearly enough to penetrate the entire brain. So although it was successfully non-invasive, it was not a perfect model, and I saw that there was still a lot of optimizing that could be done. And what I did with this information is I tried to combine the two methods. So what was good about the SOUL model was that the opsin didn't require a lot of light to be activated, but the problem there is that it was activated by blue light, which can't penetrate the skull too well. And then in the carmine model, the type of light was actually able to penetrate the skull really well, but the opsin wasn't good enough at receiving that light and becoming activated from distances farther into the brain. So what I thought of the logical solution was that I could actually figure out how to edit the SOUL opsins to become receptive to red light so that you can combine the things that are good about each of those two models. And the way to do this was to edit the protein specifically with aspects of a red light opsin that make it receptive to red light. So what I had to do first, and I did this part in Benchling, which is an online molecular biology software, is I found an opsin called channel rhodopsin, which I'd been speaking of before because unfortunately there were no SOUL opsins available, but SOUL opsins and channel rhodopsin are similar because they're both receptive to blue light, and that was the significant property in this experiment, so it worked similarly well. I took the channel rhodopsin as a whole and isolated the kinase, or the smaller piece of the opsin, which made it receptive to blue light, which was the channel rhodopsin kinase. And then I took a few different opsins receptive to red light. So I took one called Crimson, and then Carmine, which I was just speaking about. I took each of them and I isolated their respective kinases, which make them receptive to red light. And after meeting with some experts from big universities such as Harvard and MIT, because I personally wasn't an expert in protein engineering or gene editing prior to this, I had some background knowledge, but I definitely wanted to get more knowledgeable in that field before I performed the experiment. And I learned that when editing different proteins, it is really challenging to ensure that the protein doesn't break during the process. And one way to make the protein less likely to break through that process is by making sure that the protein you're editing into the initial protein is very similar. So I wanted to figure out which of those two red light proteins was more similar to the protein receptive to blue light so that I can ensure that I didn't break the protein. So I isolated the DNA of the blue light protein and then through benchling ran an alignment, which is essentially when you line up the DNA of the blue light protein with each of the red light proteins and see which one has more similarities. I'll put the different alignment results on the screen right now, and what you're seeing is all of the red visible is parts where the two sequences do not align. And what you'll be able to tell from seeing the carmine one and the crimson one is that the crimson one had a lot less red, and it only has that one really long red block at the end because crimson has a longer sequence than channelrhodopsin. So the red is just there because there is more DNA that continues to go, whereas channelrhodopsin finishes at that point. But overall, crimson is far more similar. So I figured out that what that helped me determine is I need to edit the SoulXChrimson with crimson as not to break it when I want to make it receptive to red light. And the way that I would execute that protein editing / engineering is by utilizing a tool called restriction enzymes. So when you cut out a certain piece of a protein using restriction enzymes, there's one strand on either side of the cutout portion that still remains in the protein, and those are called sticky ends, and they make it really easy for a new portion of a protein to latch on to the pre-existing part of the protein that's still left. So by utilizing those sticky ends, then you can make sure that the new portion of the protein is edited really successfully into the initial protein. Then my final hypothesis is that by editing the SoulXChrimson to be receptive to red light by using DNA from the kinase of a crimson protein, that I would successfully make the SoulXChrimson receptive to red light, and therefore I would be optimizing the model for non-invasive optogenetics to be able to perform it entirely non-invasively and have it be able to penetrate much deeper areas of the brain because this opsin, SOUL, requires a lot less light to be activated. So that is how I created a model for non-invasive optogenetics, and that solves a lot of problems both in the stigma aspect and in the stunted development of optogenetics, in order to, at least for the time being, create a model that's most appropriate for society today, most successful in its development, and likely to receive FDA approval, and also most widely accepted by society in order to help the millions of people who are struggling from brain-based conditions. And similar to channelrhodopsin, SOUL is excitatory, so it has especially exciting uses for neurodegenerative conditions such as Alzheimer's and Parkinson's, ALS and Huntington's, and many more. And there are millions of people who have to experience the adverse effects of these conditions every single day. And with this new model for non-invasive optogenetics, although it's not completely done, certainly with a relatively simple experiment, then the accurately edited protein which could best optimize this model would be able to be found, and from there forward, then optogenetics could be performed non-invasively and even more non-invasively than it had been in the past to enable this incredibly better future and improved quality of life for the millions struggling from neurodegenerative conditions. Thank you.